One of the things we did, we tried to exacerbate the phenotype of mice that prematurely aged. The idea was that giving them a high-fat diet would make them age faster. And it turned out we were actually completely wrong on this. Because I think to some extent fortuitously, the high-fat diet was contained a relatively high amount of medium-chain fatty acids, which is called a ketogenic. And so that kind of led us to the idea that ketones might be important.
Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hello, I'm Dr. Gil Blander. Welcome to Longevity by Design, how to live a longer, healthier life. We are produced by InstaTracker, your science-based guide to optimize your body from the inside out. Our guest today is Dr. Morten Scheibye-Knudsen, who will discuss aging research, the cellular and organismal consequences of DNA damage on age-related chronic diseases and healthspan, and intervention for targeting DNA damage. Morten received his MD degree from the University of Copenhagen, after which he started his research track as a postdoctoral fellow at the National Institute of Aging, NIH, in Baltimore, where he was for close to a decade working toward developing intervention in rare genetic diseases characterized by premature aging. He returned to Copenhagen in 2016 and is now associate professor running a lab where he applied machine learning algorithms to define aging phenotypes and develop interventions for aging. He combined computational approaches with a wet lab exploration to understand normal and premature aging, with focus on DNA damage and intervention leading to healthy aging. Beyond this scientific contribution, he is the Chief Scientific Officer at the Healthy Longevity Clinic, the organizing of the Early Aging Research and Drug Discovery Conference, ARDD, served as the chief editor at Frontier in Aging, and is the founder and president of Nordic Aging Society. The mission of his work is to discover intervention leading to healthier, happier, and more productive life. Welcome, Morten.
Thank you so much.
And I want to say that I met Morten a few times at the ARDD, which is an amazing conference. And I want to thank you from the bottom of my heart for organizing this amazing conference. We'll discuss it later, but I wanted to start with that. But maybe before we'll dive into science, maybe you can give us some background about what led you to become a scientist.
Yeah, so I've always been interested in science, actually. So my brother's a physicist, and as a child I was reading science fiction and these things. So it's always been, science has always been close to me, and I've always been fascinated in trying to understand nature and what happens around us, and particularly in the context of sort of aging research. This is something that I think many of my colleagues in the field have I think to some extent a similar experience. You see your grandparents or your loved ones grow older and frailer and become sick, and you wonder why we can't do anything about that. And so that really led me actually to get my MD. I went into medical school because I was thinking that I would be able to develop some interventions so that we could become healthier for a longer period of time. The rest is history, and hopefully more history coming.
Yeah, hopefully you have a lot more in front of you than behind you. Absolutely.
That would be fantastic. Yes.
So can you give us a high-level background of the research that you are currently doing at your lab?
Yeah, so as you mentioned, we use computational approaches to analyze phenotypes or characteristica of human aging, or aging in general. We also use model organisms. and then we use that either as a way to determine if a treatment or an intervention works, or to identify new interventions. And we do that both with animal models that we then test various things on in the lab, but we also recently started doing clinical trials. So this, I think, is a very exciting development, and I think this is really sort of the where we all need to go in the field, because I think there's a lot of we have a large amount of knowledge, I think, already about potential interventions that might work in animal models, but we really need to translate that into humans, I think.
Yeah, absolutely. And as I discussed in the introduction, a part of your work is on DNA damage. So can you explain to our listener what is DNA damage and what causes it?
Yes, absolutely. So we are in many ways, if you think of aging as your car growing old, it rusts and it gets damaged. We also accumulate damage over time, but it's of course much more complex. And so our DNA is our blueprint for the cell. It's like our manual for the car. And if the manual gets damaged, you can't repair the car, you can't fix the car. So there's no going back. So over time, we damage our DNA. We have a lot of enzymes. We have more than 400 enzymes that try to fix our DNA, little mechanics that are running around in our cells to try to remove the damage. And if we lose our ability to repair the damage, or if we get exposed to things that induce more damage, then we age more rapidly. And so part of my lab is looking at diseases where children basically age faster than they should. And these are most often caused by problems with repairing the DNA. So these children have mutations in single genes, and those genes are typically genes that are involved in repairing DNA.
Yeah. And just a background on myself, I used to work on the Werner syndrome
Okay. Yeah.
protein, WRN, which is also part of, it's a bit later because I assume that you talk about
That's true.
progeria. I spent, I would say, maybe a decade on the premature aging syndrome that related to DNA damage. So I completely understand your viewpoint. And yeah, can you a bit elaborate about those diseases, that DNA damage problem that basically cause a premature aging?
So I mean, there's actually quite a large amount of different diseases where you can consider that they have premature aging. So we published a paper, I think it came out a month or two ago, where we tried to define what a premature aging disease actually is, because that in itself is also kind of challenging, because all these diseases have sort of certain overlaps with normal human aging.
Yeah.
But many of them, or all of them actually, they don't show sort of the perfect phenotype of human aging. So Werner syndrome is one of them that actually come closest. I think that it has a very strong overlap with human aging, but there are many other diseases where you see an overlap with human aging. I've worked with Cockayne syndrome. I still work with Cockayne syndrome. And then we have a couple of new diseases that we're working on that I cannot disclose, but we have characterized in collaboration with some physicians here at the University of Copenhagen, where we at the main hospital here in Copenhagen, and we've generated knockout mice and have characterized those. But the take-home message, I think, from many of these diseases is that you really need to be able to repair your DNA. And if you do not repair your DNA, you accumulate damage, and that damage can lead to cancer, but it can also lead to degeneration, so cell death and degenerative diseases. So it's very important to somehow keep your DNA healthy as we age.
Absolutely. And how our body repair those DNA damage in, let's say, in a normal body that doesn't have age-related diseases? How do we do that?
So there's a number of different pathways that repair the DNA. It depends on the type of DNA. So our repair capacities have been shaped by the type of damage that our cells are exposed to. So some deal with small modifications to the DNA. This is a repair, for example, by base excision repair, which replaces a single tiny bit, like a screw in a way, or like a tiny button in the car, and puts in the correct button so that the car will work again. Then there are things that repair larger structural differences. So there's something called nucleotide excision repair, which will repair lesions to a single strand, but larger lesions, and it will take out a chunk of the DNA and then put in a replacement part. And then there are things that repair even larger lesions or more serious lesions where you have effects on both DNA strands. So normally we have two strands. One is a template, and one can be a template for repair. So if you have damage to one strand, you can take out that strand and put in a new strand and then repair the DNA that way. But if you get damage to both strands, that's an issue. And there are repair mechanisms that try to deal with that. So there's something called double-stranded break repair, which will try to fuse strands together, or do something called homologous recombination. It gets very technical. And then there are other types of DNA repair pathways that show various types of overlap. But I think the take-home message is that there is that for every lesion, there has been an evolution to adapt to that lesion and find a way to solve that for organisms. So many of these repair pathways are also found in other organisms, even in yeast cells or some organisms that are very far removed from us. So it's actually, I think, interesting in considering sort of what are the damages that accumulate with age, and how do they contribute. I think that's actually still a little bit debatable. We know that mutations accumulate with age in our DNA, but whether or not those mutations are driving a phenotype is not clear. They're probably driving cancer, but whether or not they're driving degeneration, such as neurodegeneration or cardiovascular disease, cardiomyopathies, for example, I think is not clear here. It may be that there are other mechanisms in play. For example, that we have damage that we cannot repair. So this is, for example, this is work from the late Judith Campisi that showed that persistent DNA damage. So you can accumulate a few bits of few DNA damage sites around your genome. So our genome is very large, 6 billion base pairs, but only 3 lesions in somewhere in that giant piece of these giant pieces of DNA is enough to drive a cell into senescence. So that what those lesions actually are is not clear, but it seems that the DNA damage response could be involved in the aging process by activating senescence or cellular or cell death. So apoptosis, driving cells to die, and maybe driving stem cells to die. So I'm very DNA centric in my view of aging, but there are many, probably other mechanisms that are causing us to age, I think.
Yeah, absolutely. But I agree with you that the DNA damage is a very important path, and there are so many enzymes that can fix it, and so many different pathways. I think that the take-home message for our listeners is that DNA damage is important. It is accumulated with the aging process. We know that. And it can cause a lot of different effects. So my follow-up question for you is, what question are you asking right now to try to understand better what is the relationship between DNA damage and aging?
Yeah, so as I alluded to before, that it appears that over time we accumulate DNA damage, and perhaps some types of DNA damage that we cannot repair. So we have a huge machinery that can repair all types of damage. And if you irradiate a cell with ionizing radiation, for example, you're going to create a large number of and a large variety in the type of damage that the cell can repair. And so 99% of that damage will be repaired. But you can measure that by looking at sort of alarm bells that go off around the genome. But in some of those areas, the alarm bells are not shut down, which means that the DNA damage persists. And what that damage is not understood at all, I think. And I think that maybe these represent sites that are biochemically where we don't have the right repair tools basically to repair those sites. So some of the stuff that we're doing in the lab is that we're trying to develop drugs that maybe does something to the repair site, and we're trying to develop enzymes, for example, that can so basically not human enzymes. We're engineering new repair enzymes that can go in and then repair the sites to remove this type of damage. So I think this is an exciting prospect. So this is some of the stuff we're doing.
Yeah, absolutely. It's definitely out of the box thinking. So I like it a lot. And your research is also, you have done some research on NAD and aging. And maybe if you can start by explaining to our listener what is NAD,
Yeah.
and
So
why is it relevant for age-related diseases?
yes, this is a good point. So NAD is an sort of central molecule in our cells. We use it for a large number of biochemical reactions as something called a redox factor. So it's basically like a tiny battery unit, you can say, and that battery unit is driving enzymes to do stuff. And so it's essential for example, generating energy in our cells. And so energy runs in the currency called ATP in the cells, and to generate ATP, we need NAD. And NAD is also so it's a cofactor basically in most reactions, but in some reactions, NAD is actually the battery is being eaten basically by the chemistry. And with age, it's been observed that in some tissues, you lose NAD with age, so it declines with age. And we observed that also in it's actually, I think, more than 10 years ago now in some of these premature aging disorders. And I remember actually clearly when we observed that, that was sort of a key moment in my career, because as I mentioned before, these alarm bells, when you get activation of the DNA damage response, there's an enzyme called PARP, which is called poly-ADP ribose polymerase, which chews up NAD. So it eats NAD and degrades it basically to create a larger, to amplify the alarm signal. And so we saw that NAD declined, and we saw that there was more alarm signal going on. And so the discovery was that this was probably driven by this activated PARP signaling.
Yeah. And we discussed in the past in this podcast about NAD and CO2, but definitely CO2 or CRT1 is not the only enzyme that use NAD. PARP, I think that PARP using maybe 100x more NAD than CO2, correct? Something like that. It's crazy. It's as you said, it's basically deplete all. Yeah,
it's a large NAD consumer for sure.
Yeah.
And there are others that are also quite large. There's something called CD38, and we've actually we have crossed CD38 mice. So this is an enzyme that converts NAD into something called ADP ribose. So it's basically a kind of a signaling molecule. And there's some work that shows that the loss of NAD you see with age may be due to increased expression of this CD38. But so we can see in our CD38 mice, for example, we still see loss of NAD in multiple tissues. So CD38 is definitely not the answer to everything. And I think here the DNA damage response may be something that drives this loss of NAD.
Yeah, absolutely. And talking about NAD, some of the precursors of NAD, like NR and NMN, are becoming very popular for, let's say, average user, based on some knowledge that are available in the literature. Um, so, are there? What is your view about NR and NMN supplement in human? What are the benefits and what are the risks of using it?
So yeah, I mean these are good questions. So I'm part of an NAD European NAD consortium. This is it's a EU funded Marie Curie consortium, and here we also discussed the context of NR and NMN and so forth. So I don't think there's much difference in terms of the NAD increasing capacity whether or not you use NR and NMN. In terms of the efficiency of NR to impact aging in humans is still ongoing work, I would say. There's work in mice showing that if you give NR late life in mice, you extend lifespan of mice. It also appears to improve cognition in mice. And so there's been some clinical trials in humans showing that, for example, in individuals with the Parkinson's disease, if you give NR, you may be able to attenuate some features of the disease. There's a recent work coming out with cognitive impairment, where there wasn't a clinical effect on the phenotype, but it was also a relatively short trial. But you did see a reduction in biological age of the individuals with the Alzheimer's disease. We have done also trials in my lab with the NR. So we've done a placebo-controlled trial with NR in both healthy elderly. It's not published yet, and in COPD patients. So this is in preprint, so people can see it. So this is in patients with smoker's lung, and the reason we did that was because when you smoke, you induce DNA damage. This is part of the pathogenesis. Probably you induce DNA damage in your cells in your lung, and that could drive loss of NAD. One of the things you see with COPD is also that you have inflammation in your lung, and I think the most commonly observed effect of NR is that you reduce markers of inflammation. So in our trial, we did a placebo-controlled NR trial and found that that NR did reduce inflammation in the lungs of the patients, and we also found that so we found actually that the severity of your lung issue correlated linearly with your NAD levels in your blood. So there's definitely a connection there, and I think so. I mean, I take it myself, but I usually caution people to take it because it is still an experimental drug. We have no idea about the long-term effects. We don't know if there is side effects that we have not thought about. In our placebo-controlled trial, and this was a 12-week trial, we actually did not see any side effects of the drug, but there could be actually serious side effects by doing longer treatment. So people should know that it's a very experimental. I think it's still a very experimental drug, even though it is a supplement.
And how big was the study? What was the size of this?
So the COPD patients were 20 in each arm, so still a relatively small group, but it was enough to see a significant effect.
That's cool. I didn't know about that, so it's good to
Yeah.
know. And for our listener, if you are really interested in NAD analogs, we had an episode with Joe Bauer a few weeks ago.
Yeah, so he's the right person to talk to, for sure.
So we spoke about it for an hour or maybe more, but with you, Morten, we want to extract your expertise in other direction as well. I know that your research also includes some work on a ketogenic diet. So first, can you explain for our listener what is ketogenic diet? And second, could you explain the effect of that on DNA damage and repair and aging?
Yeah, so this is actually an interesting and related question. So when I was in Baltimore, I and did my postdoctoral work. And so, what are ketones? So ketones are basically molecules that are synthesized in the liver when you're fasting, or if you eat a diet where there are very few carbohydrates but a lot of fat. And so this makes your insulin levels low. You have high glucagon, and that induces the liver to start synthesizing something called ketones. And so the reason it does that probably is because the brain cannot metabolize fatty acids. So the brain needs some fuel. So it will utilize glucose that's synthesized in the liver, but it's not enough. So it also needs additional energy, and that comes from these ketone bodies. And so it's basically fatty acids that gets broken down into small pieces, and then those pieces can go to the brain, and the brain can eat those pieces. And one of the things we have been working on in relationship to the ketogenic diet is this idea that so when you have DNA damage and you lose NAD, your metabolism becomes affected. One of the things that happen is that you will produce more lactic acid. And lactic acid is actually a molecule that increases with age. So if you look at tissues, you produce more and more lactic acid. This is the same thing that happens if you run really for a long time and you don't get enough oxygen to your muscles, then the muscles will not be able to break down the glucose, and then you produce lactic acid instead. And so with age, you actually begin to produce more and more lactic acid. You cannot metabolize your glucose properly. And one of the possible reasons is also because a potential reason is that you lose NAD, and then that drives a loss that drives the production of lactic acid. So ketones kind of circumvent this and can be used as an energy substrate independent of glycolysis. And so a hypothesis that we've been working on, and that we're still working on, is that ketones can be used this way. And in addition, we also know that this is work from Eric Verdin showing that ketones can impact the epigenetic landscape by changing how genes are expressed, and our genes are controlled by many things. And one of the things that happen when you get DNA damage, then you change the epigenetic landscape locally. And we think we have some evidence of that. It's under review, but we have some evidence of that ketones can go in and fix some of the changes that occur at the DNA damage sites.
And what is the advantages of a ketogenic diet? Like, are there advantages in a specific diseases, or what are the advantages for longevity? You showed once, you described the experiment that you have done, that they live longer. Can you elaborate about that?
Yeah, so there's work on ketones and aging showing that they live longer, and the reason why that happens is not entirely clear at the moment, actually. So it's probably multiple things. One of the things that happen during fast is that your blood sugars become low, and then you reduce your growth hormone levels. You try to maintain your stem cell pools. You activate something called autophagy, which will degrade your bad proteins so that you become more efficient. And ketones itself also seem to have some effects. And there's some work showing that ketones can activate sort of antioxidant responses by modifying this epigenetic landscape. But how they actually maintain or improve health with age is not clear. Originally, a ketogenic diet has been used on people with epilepsy. And there's also work showing that it's beneficial in some neurodegenerative diseases, potentially Parkinson's disease. And it's kind of an interesting observation because this epilepsy, the epilepsy findings, I think actually, I spoke to one of the sort of world-renowned leaders in neurodegeneration, Mark Madsen, about this. So when you fast, you produce ketones, and so 2,000 years ago in the Roman Empire, even to get people to stop having epilepsy, you put them in a room and let the demons burn themselves out. So basically what happened was that you put them in the room, and then the people were fasting and they started to produce ketones, and then that broke the epileptic seizures. So ketones have been used in, maybe not consciously, but unconsciously, probably in a couple of thousand years, to actually treat epilepsy. So for the brain, it seems to be quite healthy, and probably also for the heart.
I heard some people saying that it's also good for cancer because if you use a ketogenic diet, basically the cancer cells that need to replicate a lot doesn't have enough energy, and then they are replicating less. Is that
Yes.
what you know?
Yeah.
Yeah.
So cancers, they tend to use a lot of sugar. So this is what you take advantage of when you do a PET CT scan. So then you actually have radioactive sugar that you give to people, and then that sugar will go to the cancer site, and then you can see it on the CT scan. And so when you do a ketogenic diet, your blood sugars will be low and you produce ketones, and typically the cancer cells are not so, they cannot convert so easily to a ketogenic environment, so they can suffer. And so there's work from Walter Longo, for example, showing that fasting before a treatment of a chemotherapeutic treatment seems to be beneficial. So there is definitely also something there, but I do think that we need more information.
That's fascinating. Thank you so much for all the interesting information. And I would like to switch gear and discuss the ARDD, or the Aging Research and Drug Discovery Conference that you co-founded with Alex and Daniela. And I had the pleasure to be in the last two, and I hope to join you in the next one in Copenhagen. By the way, the venue for this conference is amazing. Anyone that haven't seen it, I would recommend you to look at the brochure for this conference. And the conference is amazing. You bring the best and the brightest in the world. So congrats for that. So can you provide to our listener a bit more about the ARDD conference and its mission, and why have you decided to found it?
Yeah. So first of all, thank you so much, Gil, for the kind words. It was also a pleasure to have you, and looking forward to seeing you in Copenhagen in a few months. So the reason that we have ARDD is really an attempt to bring together all the players that can influence how we treat aging in the future, basically. And initially, this was focusing on so it stands for Aging Research and Drug Discovery. So it was focusing on pharma companies coming together with the aging researchers and trying to find solution to aging and develop interventions for healthy aging. But we have sort of over the years, so other parts of the ecosystem have joined. So we quite quickly got a large influx of VCs. So investors that wanted to actually find companies to invest, because obviously there could be a huge commercial interest in this space. And more recently, we have tried to also include a clinical community. So we have something called the Longevity Medicine Day, which is spearheaded by Evelyn Bischoff. where we try to get the clinical community into the folds of aging research and drug discovery. And the reason that we're doing this is because interventions that lead to healthy aging has a very, very small space in a normal clinician's mind. I used to be a clinician. I worked as a doctor for a year in Denmark, and I was also working in Greenland. So you never really think about sort of this type of preventative medicine, which in essence is what it is. And so we hope to be able to stimulate that part of the ecosystem as well, because if we want to bring health to the public, we need the clinicians also.
Yeah.
And then the final part of the puzzle, which is kind of emerging now, is to get also political interest. So we have a minister of senior citizen in Denmark coming to the conference. So I think, in my opinion, there are sort of two big ways that we can change the change society and change how we view aging. One is to get political change. The other is to get big pharma to develop drugs that the doctors will then prescribe. And so I hope that we can succeed in at least one of those.
As a Longevity by Design podcast listener, you understand the value of improving your health for today and for all the years ahead. And if you want to live your healthiest, longest life possible, you need to understand what's going on inside. At Inside Tracker, we take a personalized approach to healthspan optimization that eliminates guesswork from your wellness plan. Inside Tracker analyzes blood biomarker and DNA data, along with physiomarker data from fitness trackers like Oura Ring, to deliver personalized food, supplement, lifestyle, and exercise recommendations that allow you to take control and improve your health span. And for a limited time, Longevity by Design listeners can get 20% off at the Inside Tracker store. So if you're ready to receive a personal health analysis and data-driven wellness plan to optimize your body for the long haul, then it's time to start inside. Visit insidetracker.com/podcast to get started today. That's insidetracker.com/podcast to get started today.
What you said right now raised a few points that I want to say, and there are a few because it's so exciting what you are doing at the ARDD. So the first point is about the VC and the money. It's amazing to see what happened even in the last couple of years, that you have a huge fund in Saudi Arabia that's more than $1 billion, and suddenly a few billionaires decided to fund their own companies, and they are pouring billions of dollars to the market, and a lot of other investors. So I think that's one thing that suddenly happened. And even in the last ARDD, you walked in the room and you've seen a few billionaires walking around and trying to understand what is going on. So that's one exciting part.
Yeah.
The second is, let's go to the pharma for a second. So in Denmark, you have, I think that is the most valuable company now in Europe, Novo Nordisk,
Yes.
that developed the GLP-1. And by the way, GLP-1, we've seen it also in ARDD a couple of years ago. One of the scientists presented, and it's also anti-aging. It showed data that it can extend the lifespan. So that's another way. And then
He's
the last
returning this year, actually. Yeah, so he was the guy that actually discovered GLP-1.
Yeah, that was an amazing presentation. And then you spoke about a clinician, and I couldn't agree with you more. Clinician trained to basically fix something that is broken. And here we are talking about prevention. I think that all of those points that you raised are coming to my brain together. I'm so excited about that. So yeah, everyone that hearing this podcast and is interested, I don't know if you'll do it this year, but last year you also had like a live, not recording, but live transmission that people
Yes.
it will be this year as well.
Yeah. And we also have a YouTube channel where you can watch the presentations from the last couple of years. So I highly recommend that. And one thing actually that I haven't mentioned, which is actually a big news, is that the XPRIZE healthspan summit will actually be held in Copenhagen. So this is they will, I think, reveal the teams that will compete for this big healthspan prize, the XPRIZE, which is a $101 million prize for an intervention developed within the next 7 years that can reduce aging, both, I think, physiological, cognitive, and by biomarkers by at least 10 years.
Yeah.
So I think this is also an incredibly exciting development, and so they will come to Copenhagen and will be there as part of the Longevity Medicine Day. So this is definitely very exciting.
Yeah, it is. And one comment, we interviewed Jamie Justic, the CEO of XPRIZE, in the last episode. So anyone that want to learn more about that, you're more than welcome. And again, I highly recommend you to join either online or in person to the ARDD this year, and it will be at the end of August in Copenhagen in an amazing venue. Talking about ARDD, so last year, Morten, you provide a very exciting presentation, and basically what you have shown, and again, everyone that wants can watch it in the YouTube channel of the RDD, but I'm trying to summarize it. You started to look at a big population in Denmark, and looked at what features are mostly prevalent with aging. So can you discuss a bit about that, and what were your discoveries?
So this is one of the things that I think where Denmark has a bit of an advantage. So there's been a sort of history of collecting health data over many decades. So we looked at health registry data in one of our projects here, where we have tried to investigate features of aging based on pathological description. So basically, what is the pathology? And when he looks in the microscope, what does he see on the slide? He writes down, and we have access to more than 30 million of these descriptions, and then we could run text-based analyses on these, natural language processing, and try to identify age-associated features and see if we can find patterns that could describe aging in a sort of unbiased way. And because we have good health registries, we can also connect. We know actually the diagnosis of each individual that's in the database. We also know if they have died or not. We know the birth date. We can also actually, and we haven't done that yet, but we could connect it with the pharma registry data. So we have more than 1.3 billion prescriptions. So this is a large amount of data. But
Yeah.
what we found particularly in that was, which may be a little bit controversial to some people, but we looked for patterns of aging, sort of just at a very broad level. And one of the things we did see was that there seems to be some sex-specific patterns of aging. So we observed, for example, that for women, aging seems to maybe start a little bit earlier than for men, but then it goes much slower. Whereas for men, it starts a little bit later, maybe around 40 years of age, and then it goes more rapidly downhill. And then we use those signatures also to identify molecules that could impact aging, because the text-based nature, we could translate it into other databases. So we looked in PubMed to identify drugs that were associated with the signatures, and found some drugs that could impact aging. We tested that in the lab.
Excellent. And going back to that, and I want to deep dive into a specific point. So I remember from the presentation, for example, graying of the hair is, I don't know, it's a feature that the prevalence is 99%. What are, if you remember, what are the top, let's say, 5 features that are strongly correlated with aging?
Yeah, so graying of hair is highly correlated. Muscle weakness. I have to look it up actually. Although I give this presentation, every
I understand.
like once per week, but I never really think about reading through the list. But Those are highly prevalent features, but maybe not so clinically relevant. There are other features, of course, that are more. So facial wrinkles is one of the top 5 also.
Okay.
But there are others that are more serious, right? I think one of the things that people don't realize is that 40% of everybody will get some sort of cancer diagnosis. So this is a really, every other person, basically.
Yep.
And that's the same for cardiovascular disease. So you always think that cancer is going to hit someone else, I think this is sort of the common idea, but it's
There are 60% chance that it will hit someone else, and 40% that it will hit you.
yeah, but that's still a lot,
Yeah. No, I'm joking. Yeah. About being serious for a second, you said muscle weakness,
Yeah,
and we know. I think that that's very relevant. We
yeah,
know that we are losing a lot of our muscle mass with the aging process. So again, to our viewers, we discussed a lot about how important it is to build muscle when you are getting older and how hard it is to do it. So here you have another evidence from unbiased experiment that
yeah,
Morten and his team have done that building muscle is very important for you to live better longer. So I'm excited about it. And definitely for me, the graying of the hair and wrinkles is a good control in a way, because we
yeah,
know that someone that
yeah,
getting older. So
yeah,
I think that that's
yeah,
definitely a good positive control
yeah.
for us.
And I think I,
Sorry, you wanted to say something.
I think it's also interesting because I mean there's a lot of there's really a lot of there's a huge effort to find biomarkers of aging, right? And this is really important. But the most highly prevalent feature of aging is graying of hair. So you could consider graying of hair as a biomarker of aging, right? And if we're impacting the aging process, we should also be impacting graying of hair. So and facial wrinkles, right? So in some of the clinical trials that we run, we also use facial photographs as a biological aging clock, because it also does reflect your pace of aging. And there's definitely people that age faster and people that age slower, also based on how they look, the sort of photo aging.
Yeah, and again, if you will go to the ARDD YouTube channel and look at the Morton presentation, it started with a very nice video that show how a person age, which was very exciting to view. You also mentioned a few minutes ago that the process of aging is a bit different for male and female, and you said that if I got you correctly, female are aging constantly from very early age, while male are stable until the age of 40, and then they dip faster.
Yeah,
For me, it's counterintuitive, because I would expect the opposite, because women
yeah,
have the menopause. So how would you explain that it's actually, at least for me, it's
yeah. But
counterintuitive?
I think that's an interesting point. But if we think about this in evolutionary terms, maybe so 10,000 years ago, very few women probably reached menopause. But on the other hand, they probably did reach around 40 years of age.
Yeah.
And for men, if we're thinking about, so this is looking at pathological age, you can do age prediction and see that aging does progress in males also, right? But at the microscopic level, it's very difficult to find evidence of that. But and that points to an idea that perhaps the reason why male age is pushed a little bit further back is to allow very successful males to reproduce more, basically, because there's then room for them to spread their genes wider, so to speak. And it's actually interesting if you also look at where the evolutionary pressure comes from. So this is driven, by single mutations in our genome, and that is actually found in the male germline. So that means that the variation, the single gene polymorphisms, and the evolutionary pressure maybe is driven through the male germline more than the female. And so then it would make sense that males are healthier for a little bit longer, the successful ones, because they will then be selected for, and the unsuccessful ones, they will not matter so much in terms of evolutionary pressure. It's a bit controversial, but that is potentially what the data could indicate.
Yeah, as we know in biology, not everything makes sense at the first time, but that's the reality at the end of the day. And you also showed in that presentation that there are tissue-specific aging trajectories. So some tissues are aging faster, some are slower. Can you elaborate a bit about that?
Yeah, so this is something that we have observed. I mean, we actually don't really understand why that happens. I think that there are some tissues where you have where there could be different mechanisms at play. So we know, for example, that there are tissues that replicate a lot. So bone marrow, for example, replicate a lot. So here, things like maybe telomeres, for example, could be important there, although it's still, I think, questionable how important they are, but they could be important in tissues where there's a lot of cell growth, cell replication. But in other tissues, such as the brain, which doesn't really replicate very much, that mechanism is probably different. And we also do see differences in terms of aging trajectories in the brain and versus other tissues. For example, the brain seems to be more noisy. That could be because of fewer samples, but there's actually still, I think, 200,000 samples or something like that for the brain. So it's not a small data set, but we still don't really understand tissue-specific aging, I think. And this is something that is also an active research area in the field.
But generally, the brain ages slower than other tissues, or you cannot say that?
I think it ages slower to a certain extent, and then it goes faster. So it takes a longer while before it reaches sort of an aging slope, and then it goes faster, which I think corresponds quite nicely with what you sort of see clinically, right? That neurodegenerative disease are diseases that occur often quite late, except for the hereditary types, but sort of the normal neurodegenerative diseases happens quite late, whereas cardiovascular disease is a little bit earlier. And interestingly, if you think about cancer, this is a strange disease in an aging perspective, because cancer incidence actually peaks around 75, and then the incidence actually decreases a little bit. So once you're over the hump 75, then there's at least a reduction in cancer incidence, but then your risk of neurodegeneration, cardiovascular disease really skyrockets. So why that is, I think we can speculate on, but it's not completely clear.
Yeah, I can see it also, the point that you raised about cancer that started to decay after the age of 75. We see it also at InstaTracker with biomarkers such as LDL cholesterol, for example, that it peak around 60, and then it's starting to go down somehow. And the question is, are we selecting against or everyone that already died from cardiovascular diseases, or really it's going down because the chance to have a cardiovascular disease later in life is lower? I'm not sure that we have the answer for that, but I think that's similar to what you mentioned here, which there are a lot of enigmas that we don't understand yet. And Morten, you described that also in this study, you started to look for a compound via the PubMed, the targeting aging. And I remember that even one of them, you did some experiment, I think in fruit fly, and you showed that it can extend lifespan. Can you elaborate on that?
Yeah, so we found a drug called nintedanib, which is a tyrosine kinase inhibitor. It's used for lung fibrosis, and it turned up in the screen where we looked for kind of signatures of aging, and then translated that into PubMed and screened basically all of PubMed, which is this biomedical database biomedical literature. And so this popped up, and we tested it in cell lines to look for senescence, and saw that it reduces cellular senescence. We also saw that it reduces pathways that are involved in processes that could be important in the aging lung. And then we also saw that it extends lifespan in fruit flies.
Are you
Yes.
planning to take it to a clinical trial, or what is the next step with that?
Yeah, so this is, I think, I'm not sure if we're pursuing more than nintedanib story, because there are other drugs that are also in the pipeline. For example, Silicon Medicine has an anti-fibrotic drug that looks very exciting. So I think for us, we will probably, I'm not sure we will pursue that drug more in the lab, but we're still pursuing the registry data analysis, looking at these very large data set and seeing if we can find drugs that impact human aging. So for example, using this 1.3 billion entries data set on prescription drugs, and seeing if we can find drugs that impact aging that direction.
Sounds interesting. So also in your presentation, you use a method to look at the nuclear morphology to predict cellular senescence and aging. Can you explain to our listener what was the what have you tried to do here, and what were the results?
Yeah, so again, this actually came from trying to use some of this registry data. So before we, in the other project, we looked at text-based data, and in this project we looked at image-based data. So the original idea was actually to train an age predictor based on histological samples, so skin samples. And so this was the actual approach that we started with, but that turned out to be very difficult. And so I had this idea actually based on some of the premature aging diseases. As you know, in Hodgkin-Gilford progeria, one of the premature aging diseases, the nucleus looks very convoluted.
Yeah.
And that's been, to some extent, observed a little bit before in something called senescent cells. So these are cells that we find in our tissues that are generated, for example, by persistent DNA damage. And they are probably detrimental for the tissues in the sense that they produce these inflammatory cytokines. And so I thought that maybe we could use nuclear morphology to identify these senescent cells. And actually, I usually call them angry old men, because they are like angry old men that have lost their function and become inflammatory in their remarks
And toxic.
and toxic for the environment. Yes. So we have these cells, and there's work from many labs, Jim Kirkland and others, that have shown that you need to remove these senescent cells. That seems to be beneficial. And so anyway, so we try to develop ways to identify the cells, which has been a big challenge in the field. And so we used deep neural networks to identify the nucleus shape of senescent cells, and to be able to classify senescent cells based on the nucleus shape. So this is interesting, because in most histological samples, most tissue samples, for example, if you go to your doctor and they take a biopsy from your skin, then they will do histology on that biopsy. So they will look at under a microscope, how does the tissue look, and when they do that, you can always see the nucleus. And so we translated the work that we actually did in cells in a petri dish. We translated that into the tissues, and we could identify these senescent cells in the tissues. And so this is, I think, a really nice tool, and we're collaborating now with a lot of people to look for senescence using this tool, because it's high throughput. You can look at many slides. So we have a paper in review which is also in preprint, so you can find it on medRxiv, where we have used this on more than 4,500 breast biopsies from women, where we can see that these senescence signatures can actually predict your risk of developing cancer in the future. And so this is I think a quite significant step forward, because it allows us to risk stratify women that have gotten a biopsy taken. And we're now also collaborating with researchers and in other institutions to look, for example, at prostate, where you also take biopsies if you have an enlarged prostate and you're old, to check for prostate cancer. And here we can also run the algorithm, and that's still very much a work in progress. So I don't know if we have exactly the same signal, but it looks like there may be something. But we will definitely get back to that.
And if I understood correctly your presentation, lower amount of senescent cells make a higher risk for breast cancer. That's correct?
So this was the big surprise, actually, because so historically it's understood that senescence is a way to say that the cell needs to stop growing. It's sort of an alarm bell that signals the cells, now you should not grow anymore because you're damaged. If you grow more, bad things could happen. And one of those things is probably that the cell can transform into a malignant cell and develop cancer. So that has been known for quite a while. And because we had access to the Danish healthcare registries, we can go in and look at all kinds of diagnoses and see what happens to these people that have very high senescence and very low. And this was initially based on skin biopsies, where we can see if you have low senescence, you probably have an increased risk of developing cancer in the future. Whereas if you have also, and that was also seen in the breast biopsy study. So we see that a low signal of certain types of senescence have a really strong signal in how to risk stratify women.
Yeah. For me, it's actually not surprising, because you have less senescent cells. You have a better chance for some cells to escape from the control of for getting scientific P53, P21, and so on, P16, and basically escape from that and become cancerous. I think that what is exciting about what you developed is that this can be an easy tool for a clinician to look at slides from a cancer patient and basically assign a risk score. This is high risk, this is low risk, just based on the amount of senescent cells, which is make their life much easier. And I think that is bringing. So I think that sorry.
This is the exact idea, and for now it seems to work for breast biopsies, and hopefully we can extend that. But I think also this is trained on data before individuals develop cancers. So that means that it's something inherent with that person's ability to generate senescent cells. So it's possible, although we don't know yet, but it's possible that you can use a skin biopsy and then predict, completely, like, your overall risk of developing future cancer. Somewhere else, not in the skin, but somewhere else, because it would tell us something about your personal propensity to develop senescent cells.
Yeah. So the question is, Morten, why longevity medicine is so important for a normal clinician to add to his practice and not only fix what is broken, but also go to prevention? What is your viewpoint about that?
Yeah, so I think one sort of great example that I usually bring up to clinicians, which I think sort of opens their eyes to aging and how that impact health, is the example of cancer. So if you look at the risk of developing cancer in the future based on your behavior, so if you, for example, if you're smoking, if you are drinking, if you have a bad diet, if you are overweight, your risk of developing future cancer will increase by maybe around 76%. But if you go from being 10 years old versus when you become 60 years old, and so look at the risk of developing cancer in the following 30 years, when you're 10 or when you're 60, then the risk of developing cancer increases by 3,500%. Wow. So it's much worse, in a sense, to become old than it is to smoke. We know that smoking cessation, is a really strong way to make people healthier. But if we can somehow target processes of aging with behavioral interventions or other types of interventions, then there is, I think, a huge potential to impact people's health. So I think that this is something that really should be very important for clinicians, because we want to impact health. We want to make patients healthier. We want to make people healthier for a longer period of time. And the only way to do that is to understand the aging process and try to intervene in how we age.
Yeah, and I think that I see it in your viewpoint, but maybe in a slightly different viewpoint. The 3,500% is crazy. So I 100% agree with you. In other hand, the 78% is also important, and that's part of the prevention. And for me, longevity medicine or longevity clinic is also about prevention. So don't smoke, don't eat junk, exercise, and all of that. That's something that if you go, you are a clinician, so you know that. The clinician won't tell you that. He might tell you, yeah, go and exercise, but how would you know what to do? It's like
Yes.
It's like telling you, okay, I want you to meet with you on the moon tomorrow. Yeah. How can I get So,
Yeah.
well, I think that that's another part of that, those longevity clinics are more like prevention, and they will explain to you and give you tools to do what you need to do. So I think that both of them are very important point, and definitely the addition of the longevity medicine day to ARDD is a great addition. And yeah, I wanted to follow up with you. We discussed a lot today, and it was a fascinating discussion. And you mentioned a few drug or supplements that sounds like an exciting addition. For example, the NAD analogs. But if you look at it from a 30,000-foot view, what are, in your opinion, the most promising pharmaceutical that are now developing in development process to extend the human health span and lifespan? What is, in your opinion, I don't know, the one or two most exciting one?
I mean, I think this is a great question. I actually think that the data emerging from some of these GLP-1 receptor agonists are very exciting. So these are the skinny jabs that you take, and then you basically start fasting, and you lose weight, and then you reduce your risk of developing cardiovascular disease, dementia, diabetes, obviously. I think these are really actually very fascinating approaches, because we know from animal models that these are really one of the only ways that we can sort of impact aging, sort of in a very consistent fashion, is to impact dietary intake. So these drugs are very important. One side effect of these drugs is that you lose muscle mass. So when you fast for an extended period of time, and when you take these GLP-1 receptor agonists, then you lose your you don't really feel like you need to eat anything. So you fast, and that means that you will lose muscle also. And this will have can have serious consequences. So I think discovering drugs that can maintain muscle mass, or being able to maintain muscle mass while you fast is very important, I think.
Yeah. And in the meantime, go to the gym if you're taking
go to the gym. Exactly. I think so. So we've actually done also experiments looking at fasting in humans, in elderly humans, and I always advise individuals that they need to go to the gym and they need to do strength exercise, which seems to be particularly important when you get older. There are others, of course. The standard, I think metformin is really interesting, and I think there's more data coming out on metformin that will be very interesting also in human trials. And then we have, I think there is around 20 trials in rapamycin rapalogs, which will be interesting to follow. My fear is that these compounds may have larger side effects that could make them unsuitable for large human trials. I think senolytics are interesting. There's, I think, currently 40 or so clinical trials on the way with senolytics.
Yeah.
And of course, then there are supplements like NR and NMN, which may or may not be a supplement depending on what country you're in. But I think they will have an effect, but I don't know if they will have as pronounced an effect as some of the medications that are coming. But I think what's very exciting is that we're getting there. You know, we published a paper a few months ago where we tried to collect a number of clinical trials using either neuroscience as an approach to the trial or actually targeting aging, and there's more than 130 trials on the way. So this is a huge step in the field that we are now actually trying to do clinical trials and attempting to impact aging in humans.
Yeah, and I think that if you go a decade back, maybe there were zero. I don't know, maybe one.
Yeah.
So
no, there's nothing.
It's a
I
big
think even
revolution.
5 years back there was really nothing for humans almost.
So it's a big revolution, and we should be so excited that we are living in the era that there are so many. And I assume that at least one or two of them will succeed. So
yeah, I think so too.
Yeah. So we have a bright future. One follow-up question about. Okay, so we are waiting, and we are in the waiting room in a way to find the pharmaceutical that will allow us to live longer. But in the meantime, we don't have them yet, or can take them, but we are not sure that they are safe. So how important are lifestyle factors such as nutrition, physical activity, sleep, and so on for health span and lifespan, in your opinion?
So these are really actually quite important. So if you look at what determines a human's lifespan, genetics has a role, but it's actually tiny. It's 10 to 20% of your lifespan is determined by your genes, which means the rest is really your behavior. So this can have a very large effect on your aging, and I think there's a lot to gain there. And we know at least some of the stuff that makes us healthier, right? So exercise is very important. I think that we have emerging evidence suggesting that fasting, so having some amount of fasting in your life is probably also healthy. So I typically fast 16 hours per day. Sleep is very important, particularly for your brain. And then there are other things like stuff that you can eat that seems to be good. So actually coffee is something that I always tell people that they should be drinking. So that's associated with a reduction in many age-associated diseases, except cancer. It doesn't seem to affect cancer, but other diseases appears to be positively affected by coffee drinkers up to, I think, 4 to 5 cups per day. And then dark chocolate seems to be also healthy, which is also...
That's my favorite.
Some people think that that's a good idea. Yeah, I highly support that. There's, eating vegetables, of course, is important. To some extent, a vegetarian diet or vegan diet may be healthy, but there's a little bit of conflicting data there. It's at least important to avoid processed meats. So this is like bologna and these types of meats seems to be not good for you. And then fish oils, seems to be omega-3, omega-6 oils have made a revival, I would say, and seems to be healthy. So there is a number of things you can do to improve your health, for sure.
What about alcohol? Being at
Yeah,
ARDD, I've seen a lot
yeah.
of us going to bar 7. That's the name.
Yeah. So we have an open bar at ARDD. So that's, there's things that are also good for you. And I want to say this before I get to the alcohol. Your
Yes,
social
yes.
life has a very strong influence on your aging, actually.
Yeah.
And in some contexts, alcohol plays a role there. But if you look at alcohol by itself, it's never healthy. Unfortunately, it doesn't appear that there's a lower limit where it's healthy. But the risk that you suffer from having a glass of red wine in the evening is very minimal. It has very, very little effect, although there is probably an effect. So I would usually tell people that they can have a glass of red wine as long as they remember to go running or strength.
Yeah, and also a lot of the effect that we have seen at InstaTracker on alcohol is on sleep. So if you are consuming alcohol, don't consume it at 11 p.m. and go to sleep at 11:30. Try to consume it earlier, and then your liver will detoxify the alcohol, and hopefully the effect will be lower. But I agree with you. Social is very important, and especially after COVID, we become each of us become living in his basement and don't see anyone, and it's bad. We need to socialize, and I think that again, one good thing about ARDD that you have so many social events to be there. Morten, my last question for you, and you provided a lot of tips, but what is your top tip for a healthy longevity or a health span that you will recommend others to do?
Yeah, so I think that the largest effect we see is actually the fasting, and I think this so skipping breakfasts in our hands, when we've tested it on elderly individuals in a randomized trial, we saw that had a very profound effect. And I think it's the same effect you see with some of these GLP-1 receptor agonists, that people simply start fasting, and then you reduce your risk of many adverse events with age. So I think fasting is probably the most important, but when you are older and you fast, you have to exercise at the same time to maintain muscle mass. So fasting in itself can be probably detrimental when you're very old, and there you have to do some sort of exercise at the same time. So that's my advice.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by Inside Tracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out, using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit insidetracker.com/podcast.